All right, welcome to the last session. We are going to have Dr. Barbel, who is going to talk about large data sets and the complex models, a view from systems biology. Barbel?
Okay. Yeah, thank you very much, and thanks for inviting me. So this is actually the third conference in a row where I've got to do the last talk. The first time I had to do it, I asked my colleague, what do you expect if you do the last talk? And he said, I wouldn't know, I've never been to one. So anyway, you've got to wait for your bus, so have no escape. Two of my colleagues already have done quite a bit of legwork for me. This talk is about giving you an overview about large data sets and complex models, a view from systems biology, and maybe I should add this is a view that we kind of have in our group at Warwick. So this is joint work. Our group has, there's Sylvia, who's here, and Simona, also here. They both have had posters during the lunchtime. Kirsty is a PhD student who left this summer. She graduated. Hiroshi, David, and George are postdoctoral assistants. And then David Rand is a mathematician, and he is the director of the Systems Biology Center. So I'm with one leg in statistics and one leg in the Systems Biology Center. There's also quite a lot of experimental collaboration. So the data I show here, some of the data comes from a group in Manchester who works on the pituitary gland. I've been recently very lucky to start a collaboration with an oncologist who has come to Warwick. He's from Paris, and he's working on chronotherapy. Also, I started a collaboration about a year ago with Mick Hastings' group in Cambridge, and I will show some of his data. Some of the data that I show comes from the Presta Consortium, which was a big project funded by BBSRC looking at stress in plants. So all of the data that I look at, or that we tend to look at in systems biology, is concerned with gene expression. So you've already heard about this here. So imagine a gene is a piece of DNA. So that's sitting in the cell, coiled up in the nucleus of the cell, and it sits there like an alphabet. And a gene is a particular segment of the DNA that codes for a protein. Okay, so there is the code for a gene, which is what you get when you look at this bit of DNA. Okay, and it's these usual letters A, C, G, and T. And so normally, so the DNA itself is not very active. It just sits there like an alphabet. And what we are really interested in, if genes become active, that is when they become expressed. So when do they become expressed? Well, what happens then is what's called transcription. So the piece of DNA gets translated by a complementary strand is built, which is called messenger RNA, and this then travels out of the nucleus, and it does things in the cell. Okay, so for instance, these messenger RNA then can be translated into proteins, it's the proteins that do the things in the cell. Okay, so these two processes are called transcription and translation. Okay, so transcription is making the messenger RNA that basically takes the code of the gene out of the nucleus and produces the proteins, and or is responsible for the synthesis of the proteins, and the proteins are then the the interesting molecules that are responsible for everything that's happening in the cell. So when we talk about gene expression measurement, then we mean actually measuring the amount of mRNA. So if there is messenger RNA around, that means that this gene is active. Okay? So this is the same if you write this down as a model. So this is the standard model of gene expression. You've got the DNA sitting there, you've got transcription, and you could think of this happening at some rate that we call beta, and making mRNA. So mRNA are molecules, they can degrade, or they can be responsible for translation into a protein. And again, proteins are molecules and they can also degrade. So this is the standard view of how gene expression is happening. So this is just a graph. Now you can go further and you can write down equations for it. So mathematical biologists have been writing these equations for a long time as a standard ordinary differential equation. So you've got two populations, mRNA and protein. mRNA, so this is the change in mRNA, and you've got these two processes. One is transcription, so that's the making of mRNA, and you've got degradation, so that's the mRNA getting lost. And the rates here, the making of it is at rate beta t, and the degradation happens at rate delta. And then mRNA is translated into protein, so you've got the synthesis of protein here, and you've got the degradation of protein. So you've got four reactions which summarize the standard model of gene expression for a single gene. Okay? What have I got on my next slide? So people have been modeling this as ODEs for quite a long time. As a probabilist or statistician, you would look at this more from a stochastic point of view. So you can also look at this from the point of view of having two populations. They are random variables, mRNA and protein. You've got four reactions: transcription, degradation of the mRNA, translation, and degradation of the protein. Each of these make a change in the populations. And then you can proceed in the usual stochastic way. You write down the, you can write down, so these are the effects of these populations. Then you have the different rates at which these events are happening. And you can write down what's called a reaction network, which is a continuous time Markov jump process, and they satisfy the Chapman-Kolmogorov equations. Usually chemists call these master equations, and that describes the evolution of the probability of having, the mRNA population being equal to some value and the protein population being equal to some value. So that's just usual stochastic process stuff. And the interesting thing with this system is that usually that there exists an exact numerical simulation for these reactions. So you can exactly simulate from the system. So that's the so-called Gillespie algorithm, although it hasn't been Gillespie who was the first one who used this. The same algorithm has been used for epidemic models much earlier. You can exactly simulate these reaction networks, but the master equation, so this describing this probability here, is rarely tractable. Okay, so you can, which means that you rarely arrive at an explicit formula for any exact likelihood. And so you, if you want to do inference for these systems, you haven't got an exact likelihood, which is quite interesting. So this is just an example to show that you can do simulations for such a system. So this is the behavior of an ODE, and here if you do simulations with a Gillespie, you can see that, well, for large populations they behave like an ODE. The smaller the populations get, the more random the paths look like for these little dynamical systems, if you like. So this was just, so this is an ingredient that's modeling the behavior of just one gene. Okay, so we've got two equations for one gene, 4 reactions. Now you could bring in several genes. So this is what we mean when we talk about gene networks. So this is a little example here. In this plot, say you've got 4 genes. Where are they? Gene 1 gives rise to protein 1. Gene 2 makes protein 2. Well, protein 2 and protein 1, for instance, can form a complex, and this complex may bind to the to a region in the DNA where gene 4 is transcribed. So this region is called promoter region. And gene 3, for instance, also makes protein 3, and that might bind as well. So these two guys, if they bind to this, then gene 4 makes protein 4. Okay? And they might bind to it. These effects may have different effects. This one may be activating, maybe having an activating effect. This complex might be having a negative effect. So you can already see that with only this little example, you can get quite a complex behavior of a couple of genes interacting. Now, if you Google and you look at gene networks and you do the maps, you get pages and pages of maps that look like this. So where people are trying to model the interaction between many genes. So usually this is, very little is known about how many genes interact and which genes interact, and this is very much at the heart of systems biology, is trying to find out how these networks look like. So the example that I'm in particular interested in is the circadian clock and the mammalian circadian clock. Well, this is actually a system where quite a bit is known. So it's essentially an oscillator which has at its core involved six genes. This is a schematic picture of the cell. So and this grey one is the nucleus of the cell. And this is to show that there is actually 2 feedback loops. So at the center here, you've got 2 genes called PER and CRY. They are expressed during the day, and then they go out of the nucleus and they make proteins and do things. And sorry, actually, this is the messenger RNA. So they go out here and make protein and do things, and then eventually the protein gets into a form that can re-enter the nucleus. So that's what they do here, and they form a complex, and this complex can actually inhibit their transcription. So both PER and CRY are activated by another complex here. And you can see their own product is turning this production off. Okay, so this makes an oscillator. So this is the main oscillator of the mammalian clock. But there's another oscillator here, which basically was shown to have a have a robustifying effect on the clock. Because you have, so this complex here, there is 2 other genes, RAW and reverb, that basically have an influence on the gene BMAL, and BMAL is part of this complex here, which tunes this oscillator. So, so this is another way of trying to fine-tune the major oscillator, which is this one here. So quite a bit is known about the circadian clock, also in plants. So this is a system, or an example of a system where you know quite a bit more. And in fact, the latest model has, just to give you an idea, I haven't actually written all the equations. The latest model has 20 ordinary differential equations describing 20 state variables. Some of the parameters are known. If you look at these equations, you can recognize that they all have the structure of having birth terms and degradation terms. So you can actually write quite generic equations for the dynamics of gene expression. And lots of biologists now spend trying to understand these equations and when they oscillate and what kind of oscillations they can produce. So this is the kind of mathematical biology side. So this is a bit about modeling of gene expression, just to give you an idea that there is actually some mathematical modeling behind it that we can, so some structure behind it and mechanistic. There are mechanistic ideas for modeling gene dynamics. So what's the data? Well, we've heard earlier about microarray data. So microarray data is one of the most important data sources. It gives you ideas about the expression of all the genes in the genome at the same time. And I've been very lucky because the data I had access to was actually very nice time series data. So we had lots of microarrays over time. So you have a probe, say the cells of a person, and this may be a person who's ill, or these may be cancer cells. And the RNA is isolated, and then you make a reverse transcription, and then you hybridize it to a chip which has basically probes of every gene in there, and then you can measure the light and you can basically see how much this gene is expressed. So the kind of data that I looked at in this project here, so this is just a subset which is only showing the circadian genes. So this was a plant actually. And you can see the data was, so these microarray data was taken for 2 days roughly, every 2 hours. And these are a couple of hundreds, which are just the circadian ones. So one question then coming from this microarray data, so the biologist said, oh, well, we've got lots of time series, you can see the dynamics, so maybe you can say something about which gene interacts with what. Well, it's quite hard actually for circadian genes, because how do you know? They oscillate, so they all correlate. So there's that's a slightly problematic thing to look at. On the other hand, we had not just data for one experiment, but we had data across several experiments where the plants were stressed under different conditions. So this is an overview of what we've done for this data in order to find hypotheses for gene networks. So the scenarios as follows. You have replicate time series, microarray gene expression data. So these were time series, different time lengths, different experiments. The different experiments were covering different stresses to the plants. Okay, and so What we also had, so some others in our group had done cluster analysis and had, there's also, Chris has worked on a very nice clustering algorithm, which is very much in use in our group. Other people had done lots of cluster analysis. So we had some ideas which genes behave similarly across the different experiments. But we wanted to have a bit of a closer look at certain networks around genes. So what we also had is we had a set of candidate parents for particular genes. So biologists had some hobby genes, and they had done some experiments to find out where these, which could be parents or transcription factors of these genes. So you could have a hypothesis like this. So this is the gene they were interested in, and it could potentially be regulated by these 6 guys. Okay, so they were all shown to bind to the promoter region of that gene. So these were the time series of the parents. So there's 3 of them, 3 parents, and this is under different treatments. This was a fungus like you have for, that you also use for a particular kind of wine, botrytis, and this was Pseudomonas. So this was some treatment with another parasite, that was a bacterium. So you can see these time series for these potential parents. There's the other set. Don't think about this one. So these are our fits to these parents. So this is how they behaved under the 3 different treatments. And we also had some priors about the number of transcription factors. And we also made a prior about which transcription factors would be more influential. So you could have them all equally, but you might have other ideas. And this one also built in the uncertainty about our fits here. So then we did a modeling approach where we assume that the transcription of a gene, so that's the rate B to T, is regulated by transcriptional activators and or inhibitors, we don't know. And so the problem then is that we have a set of candidate parents. So say there's G candidate parents. In my example, it was 6. And we wish to draw inference about the regulation of the transcription of the child. So we solved this by devising a kind of switch model where we said that whenever the behavior of the parents, of a parent changes, the behavior of the gene of the child should change as well. So we defined an activation function, and then we said that for the parents, and then we also introduced a set of switch times, and we said that there is a switch if that means that there is a change in the transcription of the child. And this should happen when at least one of the parents crosses the threshold. We don't know the threshold, that's something we need to estimate alongside. So we split the total time interval into sub-intervals, okay. And then so we basically try to fit this kind of model. This is now big population data, so it's not a big problem to work with ordinary differential equations. So we assume that you've got different transcription rates, but the same degradation rate. And this one here changes whenever the parents are in a certain regime. So say parent 1 is above the threshold, parent 2 is below the threshold, parent 3 is below the threshold. So you've got 2 times 2 times 2, or 2 to the power of 6 possibilities if you've got 6 possible parents. So this here is giving you that partition of the parents where they are in a particular state, and here they are in another state, and here they are in another state. And the question is, how many states do you need? How many parents do you need? So this is the dimension of this is not clear.
Are the parent labels part of the missing data? I mean, which is my parent? Is that something you know?
Yeah. So no. Well, we use the reversible jump MCMC where we invoke different parents. So we try. So what we did is we used a Bayesian approach. We developed a reversible jump MCMC to infer the plausible models for the parents. We had a likelihood which was relatively simple. We started one. So you've got the ODE solution of the system, and we started with homogeneous variance, but it turned out that you need to allow for larger variance when the measurements are larger, which is not surprising. The algorithm has become very fast because we could write down the ODE solutions because it's piecewise linear ODE. You can basically write it down as a linear regression with an inhomogeneous variance. It becomes a weighted least squares solution, and we use that as part of the algorithm instead of doing a full Bayesian regression model, and this makes it very, very fast. So this is the outcome, for instance, for this particular scenario. So this is just to show that there's also parameters to estimate the degradation rate. This is the threshold levels for the different parents. And maybe the most interesting graph here is that, so this shows you that the model jumps between different, the reversible jump MCMC jumps between different states where there's different number of transcription factors. So most of the time we have a model with 2 transcription factors. Sometimes we have 3 or maybe 4.
Could I just check, is it the number of transcription factors, or is it like which specific
Well, which one you can then see here. Okay, so this is the transcription factor set, because you need to be careful about that, that's right. So this shows here that most of the time it likes a model with transcription factor 3 and 1, but it also occasionally jumped to this with a 4. This is 4 transcription factors here, which involves number 6, number 4, and number 2, and number 1. So this is basically additional switches, which is a nice thing of this algorithm, because you don't only learn possibly the best model, but you actually learn all the alternative models, which I think is a very important part of the information that you can give to the biologists, because they might actually say, well, the second best model is probably more plausible. So it's a good idea to actually see all the different models and how plausible they are with this data. So this particular one, you find that the best model was one involving these 2 parents. This was actually the child gene time series, and this shows the fit of our model. So you can see that under botrytis, you invoke a switch here, which is caused by this parent crossing the threshold. And you also get a switch down, which comes from this one going down again. Okay, this one doesn't do anything in this particular condition, but in the next condition, this one goes to a higher level, and in fact, then this causes this switch here. This one doesn't do much here because it's below the threshold, but it crosses the threshold here. So this causes this switch, but because they both do, so normally if this hadn't gone on, this switch should be looking like that switch, but because this one goes on as well, the jump is not quite so big. So you can already see that with 2 parents, you can get quite complicated models. At the moment, we're trying to figure out we've been estimating this for about 35 networks at the moment, little networks that biologists have given us. You can see it's really good to have an algorithm to give you this solution, because by eye you can't possibly get this anymore. At the moment, we need to think about how to translate this into writing down properly the networks for these. But it should be possible. Right, how much time have I got? Time is up, okay. So I'm not going to talk about this. And maybe just go to the summary. I didn't think it would be so... I didn't think it would be taking so much time. So maybe just to summarize. So what are challenges for statisticians in systems biology and systems medicine, I should say. So we have complex and large networks of genes and their products, as you've seen. The data we look at comes from many sources. And we have to ask many questions like, have we got replicates? And if so, what type? How many cells? Usually if we have single cell data, but this is across many cells, then we want to build in some kind of assumption that the system in the cells is relatively similar. So we found that Bayesian hierarchical modeling is a very good approach of doing this by putting a hierarchy on the parameters. So we've made good experiences with that. You can have quite messy scenarios about previous information you have about parameters. So you have to build in prior distributions. Another thing that could happen with your sample is that the measurement might have been taken and then the leaf was used up, or the probe was used up. So you have destructive sampling, which is a different system than if you observe a cell over time, which is always the same cell. So there's lots of details that you have to look at when you do your modeling, and each of this can completely change your statistical approach. There is an amazing amount of times of experiments, and they all imply different modeling approaches. So I've shown you an example with ODEs, but we've equally worked in particular for single cell data with stochastic systems, and they give rise to a lot of very interesting statistical methodologies, which really need methods like that Gareth Roberts and the like are developing, and really push MCMC to the boundary. Other typical problems that arise and that are interesting to work on is that, as you can see, you've got big networks, but you never observe all of it at once. So you always have unobserved variables. And how do you model around this? We've got some experience by using delay functions, and you would have seen maybe some of this in Silvia's and Simone's posters. Parameter identifiability is a problem if you want to try and do inference about these systems. There's the whole area of spatial temporal modeling, which is completely undone. So there's a vast amount to do there to combine temporal modeling with spatial modeling. And the whole idea of collaboration with experimentalists is completely different to what I was used to when I started the subject, because it's not just that you get passed on a data set and then you can use it as an example for something. Very often biologists don't quite know themselves what they can achieve with their experiments. So we have to very often discuss together as to what we can actually do, and very often the collaboration is like from scratch, because you need to think about what you can do in statistics. They need to think about what they can do in what type of experiments can they do, and how can this go together so you get one level further. So very often all of this is completely from scratch. And which can be very frustrating, but most of the time it's very exciting. And I've got a couple of open research questions. I'm interested in more research about the circadian oscillator, in particular under diseases. And so with my collaboration with Francis Lévy, he's a chronobiologist, or he's an oncologist working in chronotherapy. So the idea is that you have in cancer cells the circadian clock is destroyed. So therefore you have optimal times at which you can give very toxic chemotherapy, where you can minimize the side effect on healthy cells. And you can basically, because cancer cells haven't got a clock, you can hit them anytime, but you can minimize the side effect. So that's the idea of chronotherapy, is to give medication at particular times. So I'm very interested in signal processing of this type of data, where we've got data on the patient's rhythm activity, which changes under treatment, and to try and predict what is a good time for giving the drug. So that's, and then there's also a very nice spatial temporal data set that I'm interested in. So this is what Sylvia started working on. This is the clock in the, this is a large data set in the brain, in the suprachiasmatic nucleus, which is somewhere between your ears in the center of your brain. And you can see that you've got oscillations of the gene expression, which was per1 or per2 maybe. So one of the central clock genes, along with calcium. And so there the challenge is to combine this type of data with this type of model, and also trying to bring in spatial modeling. So anyway, this is some open problems, many of them. Thank you. Sorry.
Any question for Barbel? Yes.
So the reversible jump MCMC, you know, just looking at it, it looked like there wasn't a hell of a lot of information in the data, and there were a lot of parameters. I mean, did it converge well? Was it well behaved?
Yes, that one was actually, yeah. Well, most of the times they are, yeah. But I mean, the problem is sometimes that we seem to be getting too many switches. It seems to over. So you have to make in quite a sparse, put in quite a sparse prior, yeah. It likes to overfit. But it's an interesting addition to, I mean, we don't really know how many switches there are, so.
Could you just give us a little kind of, in terms of the numbers, you know, how many kind of, what was in, you know, how many of those time series did you have? And how many transcription factors?
So for the real data that the biologists gave us, we actually had 6 treatments, but only 3 of them were most informative because you had quite a bit of dynamic. Some was just senescence, so it was just converging slowly to some hardly anything happening. But it's true, you can then actually ask the question, which of these experiments are more informative than others? And for instance, we found that the first one, which had the botrytis fungus, was very dynamic, and that was really very important to the identification. If nothing much happens, it's true you don't get, for instance, then you really have problems in getting the degradation rate, because you can think if it's constant, then the equilibrium is just the ratio between degradation and transcription. So you can't get the degradation. So it needs to be dynamic in order to. But it's true. It's an interesting question to actually see which of the experiments have most information.
Okay, let's thank Barbel again in the interest of time.
Thank you.